Hey folks, in this video we're going to be talking about DNA synthesis, describing how it operates, as well as defining a bunch of the terminologies you can see down below here. Now, DNA synthesis, the whole purpose of this is the replication of genetic information. And if we've watched the previous videos, we've talked about mitosis, meiosis. Let's say you're going for a bike ride, you go for a sweet jump, you fall off, you scrape your elbow. You'll need the process of mitosis in order to reform those skin cells. But one of the important parts of mitosis is that we're duplicating the DNA or replicating the DNA. And so that's the process we'll talk about here today. Now, there are a bunch of different terms that can get a little overwhelming and a little confusing as well, so we'll talk about and describe and define each one of those. So let's say that you have just a really sloppily drawn strand of DNA, and let's make this strand of DNA a little bit prettier. Let's say that this is a strand of DNA that you have in your body. What will happen is most of the time in your cells, there will be a region of your DNA that is very, very highly concentrated with A's and T's. So those would be adenines and thymines. And those signal to the cell or signal to the DNA that this is the starting point. And sometimes the term that's used is this is called the ORI, which is short for the origin of where DNA synthesis will happen. There is a DNA binding complex that exists. Usually it's drawn as just a bunch of balls like this. And what'll happen is this ORI will actually wrap itself around that region. So it'll wrap itself around. So this is that dark region. And what that'll do is hold the DNA in one place so that it makes it easier for the certain enzymes or the certain parts of DNA replication to find that strand of DNA and begin the replication process. This complex is called DNA and then little a. It's called the complex. So that's what the AT region will reach around. And then the part that we actually want to duplicate, we're going to call this the replicating zone. It's not the actual term. It's the one that I'm using. This is the information or the part that we're super concerned about. This AT can be easily reformed. A lot of times this is referred to as the telomere. And there is genetic information held there, but they serve sort of like this pen. They serve like the tip, where if I put the tip on there, I can't write on my screen anymore, and it serves to protect this actual pen. So the way that we're going to set up this DNA here is we're going to draw ourselves a nice little diagram. And it's going to start like this, and it's going to expand out like this. So DNA, and then it's going to expand out like this. We'll try to give ourselves lots of space here. So when you're looking at a strand of DNA, normally we say it's in this ladder configuration where we have two different strands of DNA with the opposite base pairs, and they are combined together through those nucleotides. And what ends up happening with DNA replication is something called the replication fork is formed, and it's sort of like a fork in the road where those two strands break apart, go off in separate directions, and they are then going to be set up for success. We'll have a bunch of these nucleotides now singularly by themselves, and so they need a partner. It's sort of like setting up a high school dance where every single thing and every single person in that dance is going to find a partner whether they want to or not. What we call both of these original strands is we call these the template strands. So in other words, these are the templates that we want to copy and paste when we replicate our DNA. As far as the names of the two different strands, this one here, the top one, is the paternal strand, or parental strand, I should call it, not paternal. That's the wrong word, parental strand. And the one down below, this one here, is called the lagging strand. And there's a reason that these two have their names like this. So a lot of times in diagrams, you'll see these symbols here, these 5 and then the apostrophe. And so what those actually are showing is, or the way that we communicate them is 5' and 3'. And a critical rule about DNA replication is that replication can only occur 5' to 3'. And so what you can see is for this top strand, that makes it pretty easy, that going 5' to 3', well, if we want to create, the other thing, sorry, I guess I should mention here is that 5' lines up with 3'. So if we go over to this side of our DNA here, we have this 3' end. Well, if we were to start pairing up the base pairs like this with our new DNA strand, this would then be the 5' end. And so we would go 5' to 3', and we would travel in this direction. And so that would work perfectly. The issue comes with this bottom strand here. If we try to start going like this, and we start trying to go to the left, we're going to go 3' to 5', and that will not work because of our DNA polymerase. And so what ends up happening is, in order for these strands to go 5' to 3', remember this will be the 3' end of the new molecules we're forming, they are going to form short little fragments, short little pieces, and those are called Okazaki fragments. Okazaki fragments. And we'll add that to our list of terms up at the top here. An Okazaki fragment is a short fragment done 5' to 3' on the lagging strand. And so the reason that this is called the lagging strand is that this strand will slowly but surely build up a couple Okazaki fragments, and they'll all travel in this direction. They all will be replicated in the 5' to the 3' direction. But we need to do it in small little chunks. Whereas for this top one here on the parental strand, that can all happen in one big go at it. And so that's why the lagging strand gets that terminology. Now, another thing to show you here is that at the front of this, and we're going to break this down in a bit more detail, at the front of this is a big old molecule. And what we're going to see throughout this entire diagram is that anytime you have an ASE, an ASE, that's usually an enzyme. And enzymes are good because they have a specific role that they are fulfilling. And usually what they help us do is speed up the rate of reactions. And so this green guy here is DNA polymerase. DNA polymerase. And DNA polymerase, what that does is it helps us actually find the correct nucleotides and place them in the correct location. And so DNA polymerase, its role is to synthesize the DNA molecules. DNA polymerase works extremely fast. It's been researched somewhere between 500 and 1,000 nucleotides per second. And so one of the unique things that DNA polymerase will also do is that it will have multiple DNA replications happening at once. And this is what allows you to form scabs so quickly. Is that 1,000 nucleotides per second is pretty quick, but if we talk about something with 1 million or 1 billion base pairs or nucleotides, that's really not that fast in comparison. And so what it'll do is it'll start multiple processes of DNA replication at once. So this is the general diagram of what is happening present here. There's a few other elements that we could also add to this molecule. So if you've ever tangled up headphones before, if you've ever tangled up headphones and tried to just pull them apart, you'll know that what happens sometimes is that as you're pulling them apart, as you have these two DNA strands and you're trying to pull them apart, sometimes what will happen is they'll form loops. And that can be a problem. Let's say that we are trying to break apart our DNA strand right here. That can be a problem if we have a loop because we won't be able to correctly copy this region or this loop here. And so what will happen is what's called the molecular scissors. So let's see if I can draw a nice pair of scissors. They will actually cut the DNA strands and then they'll be binded together later, but this guy is called topoisomerase. Topoisomerase, and that usually goes ahead of this replication fork. So somewhere up here, we'll draw these scissors, we'd see topo. Let's zoom in so that we can make this a little bit neater. We'd see up here topoisomerase. And again, we have that ASE ending, so we know that this is an enzyme that's happening. And so topoisomerase, its goal is to prevent loops or regions of DNA that get folded and cannot be copied. So that's the goal of topoisomerase. Now, another thing that will be happening here, let me just grab a yellow color. Another thing that will be happening is when we get these singular strands of DNA, that's a very, very dangerous place to be in. The beauty of double-stranded DNA is that double-stranded DNA is protected by having two of the walls on either side, basically protecting those nucleotides. When we get this singular DNA like this is very dangerous. It's very subject to mutations. And so what we'll see throughout these molecules is something called SSB. And an SSB is a single-stranded binding protein. Single-stranded binding protein. And what that does is it protects single-stranded DNA from damage and mutations. That's the purpose of SSB. Now, what's also happening here is there's a couple other terms that we need to talk about here as well. At this replication fork, what we will have is we will have something called a helicase. And so this helicase will be sitting right up front here like this on our DNA polymerase, and what that'll be helping us to do is the helicase will be helping us to separate. Helicase here is going to be helping us to separate those strands of DNA from each other. We'll go here, we'll add one more and call it helicase, and that'll be separating the double-stranded DNA into single-stranded. And usually it's carried along with this DNA polymerase here. The last couple of things that we want to talk about here. Now, in order for DNA replication to start, in order for DNA replication to commence, what usually needs to happen is there needs to be something called an RNA primer. RNA primers. These guys here are the RNA primer. And that RNA primer is there to give the DNA replication a place to start. Okay, so this allows DNA replication a place to start. Now, on the leading strand, or on the parental strand, we will see that we only have one real source of RNA primer, and we'll see that that'll be right over here. With the lagging strand, with all of these Okazaki fragments, because we have to start, stop, restart every single time we make a new fragment here, we need to have this RNA primer present so that we have a location to start our DNA replication. Now, with that, there will also be something else added in here as well. And that will be the job of removing these RNA primers. And that'll be done by something called RNase H. And so RNase H at the end of everything, what that is going to do is RNase H is going to remove the RNA primers that we added in. It's going to remove the RNA primers that we add in. So that's the purpose of RNase H. Now there's a couple other terms that we need to talk about here. One would be ligase. And what ligase does is it helps seal the nicks between these different Okazaki fragments. And so we'll have in between these two fragments something come along called ligase. And it is going to seal up the different Okazaki fragments and join them together. So it seals up the nicks in the Okazaki fragments. That is its job. The last thing that we have to talk about here is called the sliding clamp. And the sliding clamp is a bigger structure. It usually goes along with this massive structure here. So we'll draw a big circle. And it has a couple of different parts. It differs in eukaryotes and prokaryotes. But in particular for us here, this sliding clamp bonds onto the DNA, and what it's going to do is hold in place. It's going to hold in place the DNA polymerase and the helicase as well as a bunch of other structures that we don't get into too much in this course. But that's the idea of DNA replication. There's a lot of different parts, and the key things to remember is that we can only go in the 5' to 3' direction. So for the leading strand or for the parental strand, we can go this direction, and that's a nice and easy process. For the lagging strand, the strand that lags behind a little bit, that means that we need to create these Okazaki fragments. And that is the process of DNA synthesis and DNA replication.